Hello, everyone. Thank you very much for tuning in. And based on the title, I'm pretty sure you're tuning in because you are a biologist, probably working with organoids and trying to, find new ways to automate the analysis of this organoid screening with organoids in multi-well datasets. Or you're probably a non-biologist, but hopefully one of my subscribers who happen to learn something new from watching my videos, even though the primary topic may be focused on a specific type of application. So thank you for being a subscriber. And for those who are not a subscriber yet, this would be another good time for you to hit the pause button, hit that subscribe button, and while you're there, find the thanks link, and hopefully you're feeling generous today. Okay, getting back to this topic, this is about organoid screening. So why am I doing this? Well, in the past, I've done a few of these bio applications, and I got more requests to cover a few more of I've done spatialomics, for example, transcriptomics, and I've done a few of these bio applications and other applications. So I got quite a few requests about expanding on these bio topics. So I found a good public data set and a paper where they published some results. I'm like, okay, so this is a great opportunity for me to show you how Python can be used to perform a specific type of analysis, in this case, organoid screening, where you have a whole bunch of organoids, as you can see from the screen, in multi-well, and on the right-hand side, you see the analysis. So this is what this tutorial is about, and hopefully you'll stay all the way to the end. And if you have any more requests, please post them down in the comments. Okay, now let's move on. I didn't intend to talk for a couple of minutes, but hopefully that provided a good background. We're not going to replicate this paper, but this is where I was just reading this and I'm like, oh, public data set, great, let's go ahead and get it. So this is where I found out about this public data set, and it's not just this specific data set that you'll find when you go to this IDR openmicroscopy.org. There are a whole bunch of different types of data sets. So if you want to play with these, and if you want to when I say play with these, use these while you're developing your Python skills, then this would be a great resource if you happen to be a biologist. Most of these data sets that I find at idr.openmicroscopy, if not all, they're all bio-focused, but they're all excellent data sets. Okay, so with that background, in fact, from this paper, if you look at the top right there, what are they trying to do? You have multi-wells. In some cases, you have 92, you have 380 or whatever, right? You have like multi-well plates, and in each well, you have organoids in this example. Multiple organoids, bunch of them. And you're doing something different between these wells, and you want to quantify the organoid shape, structure, basically even intensity. And in this case, it's brightfield, but if it is obviously fluorescence and you have multiple markers, then you have a lot more information to extract from these wells. But in this case, these are bright field images, and as you can see, like right from this image, these are all like against a bright background. You see some somewhat darkish objects. These are the organoids. So the goal for this exercise is to, for each well, again, these are time series. So each well, in this example, the data set we're going to use, there are 5 time points. So you have multiple wells. Well, in my case, I have 6 wells. And for each one, there are 5 time points, meaning time 1, time 2, time 3, 4, and 5. You have different images. And how the organoids actually grow, how organoids change their shape, and that is basically what we are after. And how do you quantify that? For example, here is a plot that actually shows, like, for each time point, how does the object area actually change? And I'll also include analysis that kind of helps you in statistically determining whether the changes between each time point are statistically relevant or not. Okay, so that's the background in this case. And what are we going to do? So here is the approach. Here's a very high level approach. We start with an image, of course, a time series image. And let's say this is time point 1. The next thing is I'm going to invert this. There are many ways to work on this, right? You can directly train a deep learning algorithm on these images to detect these small objects. These are all organoids, by the way, right? So detect these, but I don't want to train any. I want to find the easiest way to get to the result. And for that, doing some traditional pre-processing of images can definitely help you. So in this case, I'm going to invert the image. So right away, you can see that, okay, against the dark background, white objects. So visually, it's easy for us to see, oh, now I can apply a threshold or something to segment them, but threshold is not usually appropriate or accurate enough. So, but more importantly, these are low magnification images, as you can tell, because at the four corners right there, you can see how the brightness, how these areas are brighter. Not good if you want to segment these objects. Luckily, we have something called rolling ball background subtraction. You can apply that, and that is very good at subtracting these type of artifacts, and there you go. Right away, you get an image that you can work with. We still have something in the corners right there. You can change the size of the rolling ball, when you're doing this background subtraction, but again, that's up to you. But this is a very good image, and then we are going to segment them. How are we going to segment them? Well, again, there are many ways. I ended up using something called Cellpose, which is designed to segment cells, but the pre-trained model is also good at segmenting something else that looks like cells. Like in this case, these are blobs. They kind of look like nuclei, for example, and the same model can be used to segment these. Okay, so that's how we are going to, I try to use pre-trained models as much as possible because training is a waste of time in my view, because first you need to annotate and then you have to train and then you have to make sure that, okay, it's robust enough and all that. But if there's a pre-trained model that works 95+% accurate for these type of applications where you have hundreds of objects, I think that is okay. Although I should say you can customize this Cellpose for your own objects. That's a different discussion altogether. Okay, now once this is segmented, the next step is to measure, right? So there are various measurements you can do, like obviously area, perimeter, intensity. If these are fluorescence images, which they're not, you can also get like intensity ratios between different types of fluorescence markers and all that stuff. And in fact, if you want to get fancy, you can actually take all these area, all these attributes, and you can try to do some sort of a clustering or another machine learning algorithm to kind of phenotype. But that's not the type of application we're looking for. Maybe I'll do another video later on that. So plotting, of course, to make sure we have a visual way of actually looking at the data. So this is the goal. And now the question is how to interpret the results, right? So basically these are violin plots, as you can see, between time series. So all wells seem to show increasing area over time, right? So this is well number 1, 2, 3. This is B1, B2, B3. And this is time point 1, time point 2, 3, 4, 5. So each well has 5 time points, right? So as you can see, the mean area of these organoids, there are many organoids, right, increases as the time goes by. So that's true for most, and some wells it's not as pronounced as the other because B2 probably has the highest change and B1 here has the least change. Now, how do I know that this is an actual statistically significant change? Because the change as you go from one time point to the other must be statistically significant. What does that mean? That means it should be greater than whatever the mean or variance within each distribution is, right? So this is the core basics of statistics here. And for that, I'm going to use something called ANOVA. I hope you know if you're a biologist, because if you're a biologist or any researcher, you should also be good at statistics. And you should also be good at coding. That's why I'm doing this video, so you get better at coding. Okay, so we're going to use ANOVA to confirm whether these are statistically significant or not. Now you're like, what the heck is that? Well, I hope you already know what this is, but in case you need a refresher, here it is. ANOVA is basically, it stands for analysis of variance. You're looking at the variance in the distribution of your data and then your kind of analysis, using that to perform your analysis. So it tests whether there are significant differences. So you have two different populations. By the way, if you have two different populations of data, you have like t-test, for example. There are other tests to do to distributions of data. But if you have multiple distributions of data, ANOVA is a very good approach where you can look at, in our case, like testing at organoid areas, right? Are they significantly different across 5 time points? So like I mentioned, or from the name here, you can guess it's comparing variations between these time points to the variation within the time point. So within a given data distribution, how does the variation look like? And how is the variation between the different data, clusters of data or data points? And then is this variation significant or not? So that's what it comes down to. It's a very simple, straightforward concept. And again, it accounts for natural variation in your organoid sizes, because typically within a time point, you have a different variation of these organoids, right? So whatever the next time point is showing you in terms of organoid area distribution, that must be statistically different than within this distribution. That's what it comes down to. Okay, now we calculate both F value and a P value, and the F value as part of your ANOVA analysis. It means the basically the group variation is much larger than within the group variation. So that's what, I mean, basically summing the top 2 points down here, right? So a large F value, when you calculate this, that basically means between these groups, the variation is significant, larger than the variation within the group. Okay, so F value is a measure. Larger, good, smaller, not good. That's what it comes down to. And the p-value is like any distribution. You know how we have p-value, and then if it's less than 0.05, it says that, hey, the differences are statistically significant. So we look at the combination of F and p-value. If the p-value is less than 0.05 and there is a big difference in F value, then we can say that, yes, this is a statistically significant difference. I thought of giving you a Python lesson. I have no clue why I included ANOVA. I thought it's useful. Probably you guys know, and you're wondering why I'm covering this, but here you go. It can handle multiple time points simultaneously, as I already mentioned, unlike the t-test, which only compares 2 groups, right? So again, probably something you know if you are a researcher, especially in biology, yeah, because you work with numbers. And when you work with numbers, stats is important. Okay, please note that ANOVA tells you that there are significant differences, right, between these data, but it doesn't tell you which time points differ from each other. Like if you want to pinpoint that, you need some other post hoc tests like Turkey's test, for example. Again, please let me know if you want me to cover some statistical concepts as part of this Python tutorials. But I work under the assumption if you are at least a researcher, you know these concepts, or if you are early enough in your career, like a bachelor's or even master's, probably this is the time to learn more about some statistical concepts. Struggling to talk here. Okay, now let's analyze the ANOVA results. I'm showing you the results already, and then we'll get into the code in a minute. So these are the results that I got here, and as you can see, well, A1, the p-value is 7.4. It doesn't matter. It's 10 to the minus 140. We know it's less than 0.05. This is minus 92, minus 31, minus 35. This is still much smaller than 0.05. So what these p-values tell us is statistically, there is a difference between these data points. Now, when you look at the F value, I have 194, 121, 182, and this one is 43. This one is 211 and 123. They're all large values. Now, what is a large value? Why do I say they're all large values compared to what? I will explain that in a second. So p-values are much smaller, but how to interpret F values? So here are generalized benchmarks. Again, you have to put that in the context of what data you're analyzing and everything. But in general, when you look at F values, here is what you need to look for. Is F greater than 1? That means, okay, there might be a real effect. Is F greater than 4? Okay, there is a moderate effect, especially if P is less than 0.05. In our case, P is much less than 0.05. So, of course, yeah, there is a moderate effect. If F is greater than 10, there is a very strong effect. And if F is greater than 100, very strong effect. In our case, we got like 194. The only one that's the lowest is 43, which is still a strong effect. Not a very strong effect, but a strong effect. So except for B1, everything else is a very, very strong effect, as you can see. Okay, so the variation in organoid area between the time points for each well, in our case, is statistically significant. I just wanted to put these two side by side so you can see well, B1 has the least F value, still greater than 10, right? And you can see how based on the red line, the means, how they're kind of not flat, but slope is very low, right? So that explains why this is low. Now look at the highest one. Even visually, we saw like, okay, this was going from very low all the way to that high. So the no wonder we got a value of 200, but all the other ones are like, what is the B3? It's a value of 123. And A2 is 121. So they're all like above 100. So we know they're all statistically significant. Okay, enough of the statistic stuff. Now let's jump into the reason why you tuned into this video, probably to get to look at the code and my walkthrough of the code. So let's jump into the code now. And by the way, before jumping into the code itself, let's have a quick look at the data set. So when you click on the link that I shared down below, you will see the screen that you're seeing right now. And you can see there are multiple plates. And when I click on plate number 1, this one actually, in fact, I took a small subset of this. I'm not using the entire data set from here. So I just further took a small subset, but you can see there are 4 plates. Yeah. In this plate number 1, you have, 5, 6, and you have 4 here. So 24 of these plates, and these are time series. Yeah. So each one, in fact, I think I can zoom in, right? I don't think I can go through like each time point here. Maybe I can, I don't know. I just double clicked and it opened. Okay, so this is great. So you can see the original image, time point 1, time point 2. You see how the organoids are growing. Time point, let me click here, time point 3. And let's just directly jump to time point 5 right there. And there you go, time point 5. So these are like large organoids, but then some are larger than the others. So our goal is to quantify this effect over the time series. Okay. So I hope that makes sense. Now let's go to Colab. I'm using Colab because I needed GPU, and unfortunately my workstation died, and I don't know. I'm relying on cloud resources from now on. Okay, so here you go, and obviously you know that you should change the runtime type and use GPU, whatever GPU that they give. This is, I'm recording this early 2025. This is like January 2025. Probably the video will be out in February after finding some time for me, to edit this and everything. But whatever the resources you get, I'll print out the GPU information. So if you want, you can check that out. Okay, so what do we need? I talked about the steps anyway, no point in talking about it. So first, the libraries that do not come by default with Colab are Cellpose and CSV Deep. Cellpose is for our deep learning based segmentation. This is why we need GPU. And CSV Deep it provides us the tools to normalize the images or normalize the data for these images. So that's why I'm installing these two. But otherwise, everything else that you actually see here, NumPy, Matplotlib, Pandas, Seaborn, SciPy, they should all be part of Google Colab. If you're doing this locally, make sure in your environment you install all of these. Yeah. Okay. So let's go ahead and import our libraries, and then let's actually make sure if GPU is activated, because we're going to use Cellpose GPU for Cellpose. So let it actually load the models, and from models we will actually get the right model that we need for our organoid segmentation, even though the model is probably designed for nuclei segmentation. It's just round blobs that we want to segment. Okay, so are we using GPU? Let me make sure. Yes, GPU is activated. Yes. Now I defined a whole bunch of functions. Let's quickly walk through these. This is nothing but creating our output directories. Because I'm on Colab, I want to make sure I have everything stored not in a temporary location, but on my Google Drive. So that's why I have this. Even otherwise, it makes sense to have that. And then saving the figures, I was originally displaying it on the screen, but I want to make sure I'm saving everything so we can go through that later. That's what this is, and what resolution. So I'll call this function later on when we are about to save the images. And I did talk about rolling ball background subtraction. Again, the reason for this is when you have this non-uniformity in the background, this can be a very good tool to perform that background subtraction. So not just a regular background subtraction. Okay, so for that we are going to use morphology from, you can look at this library, the scikit image. So we're going to use filters, morphology, and measure to perform measurements later on, but morphology is coming from scikit image. So rolling ball background subtraction is part of the scikit image package over there. Okay, and obviously the result is it calculates the background, or result is our actual image minus the background. Okay, and I'm generating random colors. Am I even using this? We'll go ahead and check whether I'm using these. The original intention was to, yeah, I think we are using it because I showed you the output over there. I want to color each organoid in a random color. So that's why I'm using random colors. So there are a lot of helper functions that are not necessary for the analysis, but as humans, we'd like to visualize them before we perform any analysis, right? So that's what that is. And again, the color mask is also for visualization. And basically here is where we are using the random colors part. This random colors is supporting this function down here. And estimate the diameter. In fact, this is more like why did I call this estimate? Because we need to estimate this diameter as part of Cellpose. It helps to provide an approximate diameter, because Cellpose, even though it's a deep learning tool, it helps if you provide what the approximate size of the object is. So it does a better job, basically. That's so to do that, I'm estimating the diameter right here. And why did I call this estimate? Because I'm not kind of measuring it. I'm doing like very simple thresholding right here, as you can see. Basically using the Otsu thresholding, binarizing the image, and then okay. So once it's binary, go ahead and label the images. I've done many videos on this, so hopefully you know what we are doing here. And then from our measure, from scikit image, get the region properties. And region properties, actually one of the measurements we can get is the equivalent diameter, where it takes all the pixels and fits it to some sort of a circle, let's say. Not fits it to, I mean, it basically calculates what the diameter is based on the pixels. And then I'm just plotting them for visualization. And at the end of the day, what we get is basically what the mean diameter is, right? No, actually, I started off with mean diameter, but then I realized sometimes it actually gets these blobs together because we're not doing any watershed segmentation or anything to separate the joined objects. So that's why instead of taking the mean, I'm taking the median in this case. Yeah, the median diameter is more of a good estimate in this case than the mean diameter. That's it. So this entire, the point of this is, since we are automating the process, you don't want to enter the mean diameter every time you're segmenting an image or estimated diameter. So I just basically am measuring it, estimating it right there. Okay, the key step here is segmentation. So segment and visualize. So basically the first step is, hey, estimate the diameter, because this is one of the parameters that actually goes into your model. So here, you see model.eval, go ahead and diameter equals to diameter, okay? So that's one. And I also put, okay, if the diameter, you get some error or whatever, if the diameter is not there for whatever reason, go ahead and put a value of 30 pixels, because that's kind of mean between all the images, yeah, time points I have. But hopefully that's not the case in our case. Okay, so all we need to do is now define a model and predict using that model. So here is where we are defining. So it's models.cellpose, and which model we are using, the cyto model. So Cellpose has a nuclei model, it has like a cytoplasmic model, and we are using the cyto model because our objects kind of look more like cytoplasm than nuclei. Remember, when the objects are very small, they may look like nuclei, but once they're a bit larger, the edges can be brighter and there is like intensity variation within the object. So it mimics basically a cytoplasm. That's why I'm using the cytoplasm model. You can experiment with other models. Of course, we're going to use GPU. So that's how we initiated the model. And now that we have the model, we are going to evaluate using the image, and approximate diameter is right there, and flow threshold. I did a video on Cellpose, so watch that video to get more information, or just go to the documentation to learn more about what these actually mean. Okay, this is not a multi-channel image, so we are going to use the 0th channel, like it's just a bright field image, it's a single channel, so that's what that is. So one of the outputs, of course, you get flows, you get styles, and a bunch of other stuff from this model.eval, but what we care about is masks. And the mask, I'm going to apply the color mask function. Remember, we applied the color mask when you have labeled images, objects as input. It's going to randomly color it. Why? So we can actually go ahead and look at it. That's what this entire part is. And then process the image is where we are processing the image for object areas and all that. So obviously the input is going to be the mask that actually gets generated from the Cellpose segmentation. And then, process includes that step. So step number 1 is go ahead and read the file and then go through, what is the well ID here, because it's a multi-well plate, right? So look at the well, and then within each of these wells, go ahead and look at different time points, because we have 5 time points in our case. So it goes through time point 1, time point 2, and you get your image. And remember, we're going to invert it first, and then we're going to do rolling ball background subtraction. And once the rolling ball background subtraction is done, we're going to normalize it. This image, and that normalized image is going to go as an input into our segment and visualize, which performs our Cellpose segmentation. So the diameter is going to come from there. And then once the image is segmented, you get a mask, and that mask we are going to measure the region properties. And what region properties? Well, basically the object area is what we're going to get from here. Or is the one that I'm using here, but you can get intensity, you can get a whole bunch of other. So I encourage you to, get those outputs and whatever. In this example, I'm doing plotting the area evolution. You can do intensity changes, mean intensity changes, whatever. You know the drill. So that's what we are doing. This rest of this is just plotting. I'm doing a violin plot because that is the best with this type of data set. You have multiple organoids and you want to actually look where the mean is, where is the 95th percentile and all that stuff. So just visualization purposes. And then we also need to perform, I chose to perform ANOVA, which I already talked about, to quantify whether the changes between these time points within each well are statistically significant. And ANOVA is extremely simple. It's from SciPy library, the one that I'm using. And within SciPy, you have stats. And within stats, you have this F one-way, which basically gives you the F scores and the p-values when you perform this ANOVA analysis. And we are going to report that for each well. That's it. And in addition to that, I also added a heat map where we do area ratios. So what we do is take the first well and the last well, and then what is the area of the mean area in this case, as you can see, that we are calculating for the first well and the last well, and what is the ratio, and we're going to plot that ratio as a heat map. Again, there are many, many ways you can report these, and this is what I chose to do. So let's go ahead and run these. These are all functions, so they should run pretty quickly. There's nothing to execute there. And finally, let's go ahead and put everything together. And okay, right here, let's put everything together. Basically, I'm giving my output directory and the input directory where we have the wells. It's going to go through each of these wells, look for TIFF file. Again, these are time series images, so TIFF files, obviously. You can rewrite this if you have individual PNGs or whatever of your image, right? This is basically reading the images part. And yeah, that's pretty much it. There is, and we are saving the outputs into our CSV, and we are going to perform the basic analysis, and then area, and perform ANOVA and the heat map. And once this is done, you should see all the results. So I hope the segmentation part, I tested this, obviously, it worked yesterday. So I hope it works again today. And this should be pretty relatively fast, even though you have thousands of objects or probably hundreds of objects in each image, and you have 5 time series. So total, you have 25 images, right? Or how many? We have 6 wells. And so 30 images actually for it to process. And it is doing this in a pretty decent time, time 1, time 2 in this well. So I'll pause the video and continue as soon as this is done, so we can have a quick look at the results. And it's almost ending. It's well B3 time 0.5. It's about 4 minutes. So that's pretty fast actually, given what we're doing here using deep learning. Okay, so let's go through some of these. And this is exactly what I shared earlier as part of the PowerPoint presentation. As you can see, the ANOVA results 194, 121, 182, they're all much greater than 100. And 43, basically, it's less than 100, but 100 is like way different, right? So it's anything above 10 is pretty good. It tells that, not pretty good, it tells us that there is a difference between each of these data sets. What else did we do? Basically, for each well, these are basically the mean areas. So we plotted the heat map. So all the data should be saved. And I opened my Google Drive right here. And here you can see what it saves is 4 different types of results. One is the segmented image. Let's look at the processed images first. And let's look at these processed images. So these are the ones where there are background subtracted. And that's it. Yeah, these are the processed ones. So inverted and background subtracted. And then if you look at the segmented images, these should be the nice colorful ones. I chose to make them colorful because it's for our eyes. We can separate, we can distinguish colors better than gray levels, right? So that's why I like to apply random colors. But anyway, so there you go. So these are all processed. You can study them carefully. In fact, you can, if you want to adjust Cellpose parameters to get better segmentation, this is the reason why I'm saving all these, not just for fun of it. And the last one that we are, I also save diameter plots. These are histogram plots for each image. What is the mean diameter? I mean, what is the diameter spread for these organoids? But let's just look at the analysis plots and let's look at the result here. So here is the heat map. So there you go. Again, these make more sense if you have like a very large, like 380 plus, multi-well plate or even 92 multi-well plates. Then you can see these. You have much better data. Hopefully, one of the data sets on IDR has such type of large data. Okay, so I'm also saving the area evolution. You already saw these plots. So, and finally, for each object, I have all the information saved. Why? You never know when you need to do something. Okay, so I seriously hope you found this to be educational, especially since you're towards the end of this video. I hope you found this to be useful for your efforts. And as usual, the code link is down below under the description. Please do not forget to hit the subscribe button. Thank you again for sticking with me all the way throughout the video. Thank you.